Yerezalerin yüksek bağışıklık sistemi nasıl çalışır? Skin acts as the body's outer shield, constantly renewing itself to keep microbes out while also sensing the environment. When a cut or scrape breaks that shield, a repair program kicks in, not just closing the gap, but also carving out a niche where certain immune cells can settle long-term. Seeing what happens after the visible wound heals helps us understand how the body remembers past infections and readies itself for future ones. Healing follows 3 overlapping stages: inflammation, proliferation, and remodeling. Neutrophils and macrophages arrive first to clear debris and pathogens, sending out signals that draw more immune cells. Fibroblasts then lay down a temporary collagen scaffold, while endothelial cells sprout new capillaries to bring oxygen. As the matrix matures, excess cells are cleared and the scar gains strength. Each stage shifts the molecular milieu, determining which cells can remain after the wound looks closed. During remodeling, a fraction of the T cells that entered the wound during inflammation start to turn into tissue-resident memory cells, or TRM. Unlike circulating memory T cells that travel through blood, TRM lodge directly in the tissue where they first saw the antigen. This local placement lets them react much faster than cells that would have to come from lymph nodes or bloodstream when the same pathogen returns. 2 markers set these skin-resident memory T cells apart: CD69 and CD103. CD69 is an early activation flag that also holds the cell in place by blocking sphingosine 1-phosphate receptors. CD103, the alpha E part of integrin alpha A beta 7, grabs E-cadherin on epithelial cells, anchoring the T cell in the epidermis. Cells that show both CD69 and CD103 are poised for long-term stay in healed skin. Turning an infiltrating effector T cell into a CD69 plus CD103 plus TRM relies on signals from the wound. TGF-beta, plentiful in the remodeling phase, drives CD103 up through SMAD-dependent transcription. At the same time, IL-15 and type I interferons boost survival and CD69 expression. These cues rewrite the cell's gene program, shutting down exit routes and turning on pathways that promote tissue adhesion and longevity. Once established, TRM cells settle in distinct skin zones. They crowd the basal epidermal layer, where they can touch keratinocytes, and gather around hair follicles and sebaceous glands, niches rich in survival factors. In the dermis, a smaller group sits near blood vessels and sweat glands, poised to meet any pathogen that breaches the epidermis. This layout puts them in the best position to encounter invading microbes early. When a pathogen such as Staphylococcus aureus or Candida albicans re-enters the healed spot, resident memory T cells spring into action within hours. They release IFN-gamma and IL-17, which cue nearby epithelial cells to make antimicrobial peptides and call in extra immune help. This swift, local response often stops the infection before it spreads, frequently preventing any noticeable symptoms. Mouse experiments give us concrete numbers. After a skin infection clears, CD69 plus CD103 plus T cells stay detectable in the healed epidermis for up to 200 days, about 6 to 7 months, without a major drop in frequency. When those mice are rechallenged with the same pathogen, the ones with intact TRM have far lower bacterial loads and clear the infection quicker than mice lacking this resident population. Human data back up the mouse results. Biopsies from healed surgical scars or resolved skin infections show CD69 plus CD103 plus T cells persisting for several months, with markers and gene activity matching mouse skin TRM. Although sampling depth limits exact counts, the presence of these cells aligns with a lower chance of recurrent infection at the same site in clinical notes. The protective effect isn't just anecdotal. Quantitative models estimate roughly a 70% reduction in reinfection risk when a strong skin TRM population is present. This figure comes from comparing infection rates in animals with normal TRM formation versus those where TRM generation was blocked genetically or with antibodies. The size of the protection matches the observed faster pathogen clearance and milder clinical symptoms. Several factors shape how long skin TRM survive and how well they guard tissue. Ongoing TGF-beta exposure keeps CD103 expressed, while IL-15 supports steady-state proliferation. Lipid antigens presented by CD1A-expressing Langerhans cells can also give selective survival cues. On the flip side, if inflammation resolves too quickly or scar tissue grows excessively, the niche for TRM shrinks, shortening their lifespan. Clinically, knowing that long-lasting skin TRM exists opens doors for vaccine design. Approaches that aim to generate TRM, such as scarification, microneedle patches, or adjuvants that mimic tissue injury signals, could deliver durable immunity at the entry points for pathogens like herpes simplex virus or malaria sporozoites. Early trials indicate that skin-targeted vaccination yields higher epidermal frequencies of CD69 plus CD103 plus T cells than standard intramuscular shots. We should note the limits of current knowledge. Most detailed data come from animal models, and human tissue sampling is invasive, so the exact kinetics and functional diversity of skin TRM in people remain less clear. Individual differences, genetics, age, microbiome, prior infection history, can affect both the number and quality of these resident cells. Researchers are using newer tools, like single-cell RNA sequencing of biopsy samples, to map these variables. In short, after a wound closes, a specialized group of CD69 plus CD103 plus T resident memory cells remains in the scar for up to 200 days. They act as a local sentinel, ready to release antimicrobial cytokines within hours of seeing the pathogen again, cutting the risk of reinfection by about 70%. This hidden army shows how the body turns a moment of injury into a lasting advantage, turning the scar itself into a strategic immunological outpost. Every healed cut carries a quiet, vigilant guard, our skin's own memory T cells, working unseen to keep us safe.